The diversity of life. Our next unit is on taxonomy and classification of living organisms. Let's review what we've learned about cells and living organisms up to this point. We know that there's a prokaryotic cell, which has no nucleus, but does have a cell wall. Things like bacteria are prokaryotic. We also know that there's a eukaryotic cell. This is differentiated because it has DNA that's contained in the nucleus. We know that there are autotrophs. Autotrophs are things that capture light energy from the sun and convert it to chemical energy, which they use for food and which other things use for food as well. Heterotrophs must get their energy by eating, usually autotrophs or other heterotrophs. Heterotrophs include carnivores, herbivores, and omnivores. So we have the meat eaters, the plant eaters, and the organisms that eat both meat and plants. Decomposers are also heterotrophs that recycle dead organisms by breaking them down. These would be fungus, bacteria, any organism that eats dead things. So here are some examples of each of those kinds of organisms. We have your heterotrophs up here. We have carnivores, omnivores. This would be a carnivore as well. Taxonomy is the science of grouping and naming organisms. Taxonomy groups organisms based on similarities. Classification is the grouping of information or objects based on similarities. So taxonomy is the study of classification. We only know about a fraction of the organisms that exist now or have ever existed on Earth. Taxonomists give a unique scientific name to each of these species that they know about, whether it's alive today or extinct. The scientific name that they give comes from one of two quote-unquote dead languages, meaning these languages aren't spoken currently, either Latin or Ancient Greek. In the next several slides, I want you to look closely at each organism and compare their features, compare what they look like. The devil cat, the ghost cat, the mountain lion, the puma, the Florida panther. What did you notice? There are at least 50 common names for the cat that was shown on the previous slide. Common names vary according to the region that the organism is found in. So, why would we use a scientific name instead? Probably because there are about 1.5 million species that have already been named, and as you can see, the same organism can have different names just by going from one region to another. From going from one locality to another, the same organism could have different names. Scientists estimate between 2 and 100 million species that haven't even been discovered yet. So in order to clarify exactly what the organism is, no matter where it's found, we come up with this scientific name. The scientific name, or the naming of organisms, is called Binomial Nomenclature. This system was developed by Carolus Linnaeus. It's a two-name system for writing scientific names. You start with the genus. This is always written first, and it's always capitalized. The second part of the name is the species, and this is written after the genus, and it's never capitalized. Both words in binomial nomenclature are italicized if they're typed, or underlined if they're handwritten. For example, Felis concolor, or F. concolor, is the name of the cats that we saw in those previous slides. This way, no matter where you are in a region, if you refer to the organism by its scientific name, then you know you're talking about the same thing. So in Felis concolor, which is the genus and which part of that is the species? If we go by the rules set up for us up here, Felis is the genus, it's capitalized. Concolor is the species, it's not capitalized. Let's talk now about the major classification levels, going from the most general to the most specific. And remember that these are identified based on similarities that the organisms in that classification level share. Several of these classification levels have subdivisions. Linnaeus grouped organisms based on their physical similarities, but today, organisms are grouped based on evolutionary characteristics. So things that, characteristics that have evolved along with the organism over time. The major classification level starts with the kingdom, and a group at any level is called a taxon, which is where we get taxonomy from. Here are the 6 kingdoms in their domains, and I know that you have a question on some of these kingdoms and their examples. So, Archaeobacteria, this is unicellular. They get their energy mostly autotrophically, some chemotrophically. They're prokaryotic, and an example are the extremophiles, the organisms that live in extremely, extremely hot areas, or extremely, extremely cold, or extremely dark, these kinds of things. Next level, the next kingdom is the Eubacteria, also unicellular. They can be both autotrophic or heterotrophic. They're prokaryotic, which means they do not have a nucleus. And the examples we would be familiar with are bacteria like E. coli. The fungi, these are mostly multicellular. They are heterotrophic organisms. They are eukaryotic, which means they do have a nucleus. And some examples you would be familiar with would be mushrooms or yeast. The Plantae, these are multicellular autotrophic organisms. They are eukaryotic, so they do have a nucleus, like trees or grass. Animalia, these are multicellular also. These are heterotrophic, which means they get their energy from other autotrophic things or other heterotrophic things. They are eukaryotic, and these include humans, insects, worms, any sort of thing from the animal kingdom. And Protista. These are multicellular. They're heterotrophic or autotrophic. They are eukaryotic, which means they have a nucleus. And things like the amoeba, the paramecium, and algae, many of the Protista live in pond water or stagnant water. This is an example of how the 6 kingdoms are broken down. You have the archaea, you have bacteria, and you have the eukarya. Eukarya means eukaryotic cells. Under eukarya, you have the Protista, the fungi, the plants, and the animals. These are kingdoms. These 3 are domains. So the domain is a larger grouping than a kingdom. So categories within the kingdom. If you want to remember the order that these go in, remember this sentence. King Philip came over for George's sword. We start with the kingdoms. These are divided into groups called phyla. Phyla are divided into classes. So you can see we're following the sentence. Kingdom, phylum, class. Then we have order, family, genus, or the genera, and species. The species is the unique organism, the individual organism. So once you get all the way down to species, the species of any organism share the same characteristics, but there's something different about them than any other organism in the same genus. Take a look here at how the bobcat, the lion, and the shaggy-maned mushroom are classified if you go all the way from kingdom down to species. Notice we have 2 from the kingdom Animalia and 1 from the kingdom Fungi. The bobcat and the lion both are in the same phylum. They're both in the Chordata, meaning chordates. They're both Mammalia. They're both in the order Carnivora. They're both in the family Phylogeny, but here's where they differ. The bobcat is in the genus Lynx, where the lion is in the genus Panthera. And the species, meaning there are other organisms classified under lynxes, this is the Lynx rufus. The lion, however, is the Panthera leo. The shaggy mane mushroom, totally different organizational structure. It's in the kingdom Fungi. It's in the phylum Basidiomycota. It's in the class Homo Basidiomycete. It's in the order Agricales. It's in the family Copraceae. It's in the genus Coprinus. And it's in the species Coprinus comatus. So you can see how even two organisms that are similar all the way down to the end, once they get to that genus, that tells us there's something different about these two species. The Dichotomous Key is a device that's used to identify organisms. It's the most widely used type in the biological sciences for identifying things that we don't know. To use it, the user is presented with a sequence of choices between two statements, called couplets, based on characteristics of the organism. By always making the correct choice, which is tricky to do, the name of the organism will be revealed. There's a difference between using a dichotomous key compared to evolutionary classification, which we'll get to in a minute. Dichotomous keys contain pairs of contrasting descriptions. After each description, the key directs the user to another pair of descriptions, and you eventually get down to the identity of the organism. So, for example, the first set of statements is the leaf simple? You would go to number 2. If it's not simple, you choose is the leaf compound? You would go to number 3. If going to number 2, you're left with, or you're given 2 more statements. Are the margins of the leaf jagged, or are the margins of the leaf smooth? And then it directs you to the correct next statement. So, for example, if you were looking at this organism, the first set of statements, does it have 1 pair of wings or 2? Well, you can see here that there are 2 pairs of wings, so you would follow B. And then so on and so forth until it gave you the name of the organism. In modern taxonomy, you use phylogeny, which is the evolutionary history of an organism, and it's the cornerstone of a branch of biology called systematic taxonomy. Systematics, as systematic taxonomy is commonly called, is the study of the evolution of biological diversity. A phylogenetic tree is a family tree that shows a hypothesis about the evolutionary relationships thought to exist among groups of organisms. It does not show the actual evolutionary history of organisms. So these are things that have existed well before we were studying their characteristics, their common characteristics. And so we guess. Scientists take a scientific hypothesis to study how they came about. So you can see here, this is kind of that hypothesis in picture form. It starts here with this common protists. And then you can see as you go up the line, we have branches. These 2 things must have some similarities or they would not come off the same branch. As compared to sponges, cnidarians and ctenophores don't have any similarities, so sponges get their own branch. As you go up the tree, the phylogenetic tree, you get more and more complex in the type of organism. If you take a look at the top, you see the most complex type of organism would be arthropods. Arthropods and annelids, however, must have something in common because they can't come off the same branch. Phylogenetic trees, which is what you just saw, are usually based on a combination of these lines of evidence. They can be based on fossil record. They can be based on morphology, which deals with the physical structures of and organs of an organism, like what was the arm shaped like, what are the internal organs, what do they look like, where are they placed. Embryological patterns of development, which you'll see an example of in a moment. So how does the embryo develop into the full-blown organism? And chromosomes and DNA. is also a piece of evidence used in creating phylogenetic trees. Here are some examples. So fossil records. We take a look at fossils, you look at how things were shaped, how organisms, where they were found, things like that. Morphology, this is looking at the physical arms, legs, organs of the organisms to see if there are any similarities. This is morphology again. Homologous, meaning the same. So if you look at the penguin, the alligator, the bat, and the human, you can see that the color-coded bones within each structure, these would be similar things. So the green in each organism is the humerus. You can see that all 4 organisms, though we know they're very different, have a humerus. They each have a radius. They each have an ulna. The ulna is shaped differently in each organism, but it's there. They have carpals, they have metacarpals, and they have phalanges. So even though the organisms are very distinct, very different, you can see that their morphology follows a similar pattern. The bones in each arm, so to speak, follows a similar pattern. Convergent evolution. These animals have evolved similar adaptations for obtaining food because they occupy similar niches, even though they're on 4 different continents. The giant armadillo, the giant pangolin, the spiny anteater, and the giant anteater all have very similar noses because they're all eating the same type of food within their environment. So what can you infer about their phylogeny from their geographic locations? Their phylogeny would be their common ancestor. Even though they're on 4 different continents, convergent evolution leads to analogous structures, or traits that are morphologically and functionally similar, even though there's no common ancestor. So even though those 4 types of anteaters don't have a common ancestor, they have adapted very similar structures. That nose used to root out and dig out insects or ants because they need to fill that niche in their environment. Here's another example of convergent evolution. Even though these organisms do not have a common ancestor, they all have the ability to fly. Bats are mammals. Birds are not mammals. This would most likely be a reptile. So even though they don't have the same ancestor, they need similar morphology. They need similar structures in order to be able to fly. Here's an example of embryology. If you take a look along the top row, you see that you have 6 different embryos. But each embryo has very similar structure. They all basically look the same. You can see where there's an eye, you can see where there would be an ear, you can see they all have a tail, you can see structures forming along the front. If you go to the next row, you can see the embryos start to develop slightly differently, especially this one compared to this one. But at the end, as the embryo continues to develop, then you see that even though they started off very similar, they're now very different. We have a fish, a salamander, a tortoise, a chick, a rabbit, and man. Moving on to cladistics. Cladistics is a relatively new system of phylogenetics. Phylogenetics, again, is based on evolutionary history. Cladistics uses shared derived characters to establish evolutionary relationships. A derived character is any feature that apparently evolved only within the group under consideration. So it's something different about that particular organism that makes it different from all the others, even if there were evolutionary similarities.